Thank you, Tracy. And thank you, Josh, and the organizing committee for giving me the opportunity to speak here. And I'm also particularly happy because today is my 55th birthday. Thank you. Thank you. So we human beings are just bioreactors. So if you look at your gut, it's a continuous culturing system. So in microbiology, it's called a chemostat. Basically, you are constantly feeding fresh nutrients into the bioreactor, and you are also removing the cultured microbes at a constant rate. So when a particular bacteria reach a certain abundance level and maintain that level when the system reach equilibrium. And surprisingly, we have 1 to 10 times more bacterial cells in our body than our own human cells, and 10 to 100 times more genomic information, genes, in our microbiome than in our human genome. And in terms of species, we probably have about somewhere between 400 species to 1,000 species. But in terms of strains, we could have up to 50,000 different strains. So basically in bacteriology, one strain is just equivalent to one human individual. So basically, the gut bacteria, they grow in our gut so that they can produce many bioactive compounds. So those bioactive compounds can get into our bloodstream either through the normal enterohepatic circulation or very often when our gut barrier are partially impaired, and then many of these things can get into our bloodstream. So there's about 1/3 of small molecules in our bloodstream probably are derived from gut bacteria. So that's a tremendous amount of metabolites. So they can regulate our genes, modulate our immunity, and also impact our metabolic health. And well, if you look closely, when we are eating, we are not just eating for ourselves. We are also eating for our microbiome. So there are two kinds of nutrients coming to our gut and feed our microbiome. One is from our diet. So anything non-digestible or undigested, particularly I think nowadays because of overeating or problem with digestion, many people have the overeating problem, have actually many, a substantial part of the food we take into our guts probably not completely digested and not absorbed. And they will be feeding the gut microbiome. And the gut bacteria, they also have locally produced nutrients from our gut itself, like mucin and left colon cells. Our epithelial cells will fall off every 3 days. So all these dead cells will be used by gut bacteria. And there are also many secretions, bile acids and other digestive enzymes. All these things, as long as it contains energy, as long as it's some form of organic compound and can be fermented by some gut bacteria to derive energy to support their growth, they will be used by some members of the gut microbiota as nutrients. So if you are a purely vegan person and your gut microbiota still can get animal food, so that's from your own production. And so basically, I would like to focus on obesity and type 2 diabetes, and this has become a problem in not both rapidly developing countries such as China and also developed countries such as US. So obviously, there are so many studies on obesity and why people become obese. There are hundreds of series. And basically, I think we are missing something in between the seemingly driving factors of obesity and type 2 diabetes. We have something in between missing. That is the molecular chain of causation. And gut microbiota seems to be one of the missing link. Professor Jeff Gordon is the pioneer of this field, and starting from 2004, by using germ-free animals and the reconventionalization of germ-free animals, they showed that germ-free mice are resistant to high-fat diet-induced obesity. But when you reconventionalize them using normal microbiota, then the conventionalized mice start to develop obesity, even though they take less calorie compared when they were germ-free. And so they identified some lipid metabolism genes, such as one gene called fasting-induced adipose factor in the gut, which the product of that gene is promoting fatty acid oxidation and also inhibiting lipid storage. So the germ-free mice has this gene constitutively expressing at a high level so that they can only burn fat but cannot store fat. That's probably why they are resistant to high-fat diet-induced obesity. So if you knock out this gene, and then they lose this resistance. And if you introduce gut bacteria, this gene will be shut off. And only hunger, only fasting, to some extent, can turn on this gene, and then you can start to use the stored fat. And they also found that when you reintroduce gut microbiota to germ-free mice, you promote several key genes for de novo lipid synthesis in the liver, such as ACE1 fast and pipa gamma. So basically, reintroduction of gut microbiota to germ-free mice shut off genes in the gut, which are required for burning stored fat, but promoting genes in the liver for synthesizing new fat, particularly from glucose. So these are the very important findings from Jeff Gordon's group. And then starting from 2007, Professor Patrice Gagné and his collaborators, they start to another line of research by looking at LPS endotoxin from gram-negative opportunistic pathogens in animal and human gut. By using purified lipopolysaccharide endotoxin infused to mice on normal chow diet at a low concentration continuously for 8 weeks, then the mice develop obesity and insulin resistance, even though they had a low-calorie diet. So if you give this LPS endotoxin to CD14 knockout mice, no inflammation can be induced, then no obesity can be induced by this low dosage of continuous infusion of LPS endotoxin. So basically, these are all very interesting evidences showing that somehow the gut microbiota or even a particular microbial product of the gut bacteria can be contributing to obesity development. And of course, Jeff Gordon's group and many other groups, including my group, are trying to identify the particular members of the gut microbiota out of the 1,000 species or tens of thousands of strains, which would be the key players, contributors for obesity development. And like Professor Gordon's group, they published association between Firmicutes phylum and over Bacteroidetes phylum, and they see this is associated with obesity. So all kinds of signatures, structural signatures of gut microbiota has been published, seems to be associated with obesity. But then the question is, obese people have different gut microbiota compared with healthy people. Is it because I'm obese, so I have a different gut microbiota, or because I have a different gut microbiota, so I become obese? So this causality question is the first, most important fundamental question for the microbiome research field. And we know that in infectious disease field, you need to identify the causative agent for a particular infectious disease. And I remember vividly, in 2003, early spring, SARS was broken out in China and then spread out to Canada, Hong Kong, to other parts of the world. It's actually, because we don't know what's the reason, so it's very, very scary. And until this paper was published It's a one-page publication. So it said Co-occurrence postulates fulfilled for SARS virus. So when you fulfill Co-occurrence postulates, that means you identified the causative agent of this emerging infectious disease. I also argue that in this Nature Reviews microbiology perspective, we should follow the logic of Co-occurrence postulates to identify the key players in our gut microbiota for obesity, type 2 diabetes, or any other chronic diseases that you are interested. So basically, you should provide 3 kinds of evidences before you can be sure you have identified the key players, and they can become biomarkers for prediction, for diagnosis, and also become targets for prevention or treatment. So basically, you need to do a microbiome-wide association study, identify all the positively or negatively associated members of the gut microbiota to the particular disease or a particular phenotype of the disease. And then you should isolate these associated bacteria into either pure culture or into a consortium with defined membership. And then you should colonize these isolates into germ-free mice gut, for example, and then give the right environmental condition, for example, the high-fat diet. See if you can reproduce the disease. If you can, now you have a autobiotic model of your disease. And then you should understand the molecular mechanism from the colonization of the gut by these bacteria of the mice to the development of the disease endpoints in the host mice. So all these things need to be studied and understood before any microbiome research can be really translated into applications. So I'm a microbial ecologist. To me, the causality question can be converted to questions like who's there, who does what, with whom, and how, and at the molecular level. So understand who's there, you need to realize that microbial ecosystems like gut microbiota have strings as the very basic building blocks. So basically each string is just like one human individual. We know among human individuals the genetic difference is very minor, maybe 0.1% between different people in this room. We share 0.1% genetic difference among individuals of human. However, under the same named bacterial species, strings which are identified and clustered into the same species can share up to 30% genomic difference. So that means that you can we share about 10% genomic difference with mice. So that means you need to go down to string level if you want to understand the good bacteria, the bad bacteria, or the neutral ones from our gut. And we also need to understand the ecology of our gut. So basically, species are not independent from each other in ecosystems. So they form a functional group called guild. Members of the same guild, they increase or decrease together. They are exploiting the same environmental resource together. And we need to know which bacteria in our gut also form guild-like structure with other bacteria. And also we need to understand whether, like in a closed forest, the tall trees are the foundation species. They are defining the environmental condition which is fundamental to all the other members of the system. So we need to know whether in our gut, are there any species of bacteria serve the role like tall tree to the whole forest, so the foundation species. So these are the microbiological and ecological thinking in our microbiome research. So the strategy we take is not to compare diseased people with healthy people. Rather, we do interventional studies. So we change the microbiome of diseased people and see if we can get improvement in their disease phenotypes. And then we collect urine, blood, and fecal sample over time, multiple time points. And then we analyze the molecular level variations within these 3 biological windows. And then we do correlation analysis and multivariate statistics and data mining, try to identify specific signatures, molecular signatures in each of the 3 windows and correlate them and see if we can identify the key species or key members of the gut microbiota, which may be important for obesity and type 2 diabetes. And we use diet to change the microbiota because from animal studies, we are the first to show that diet is the most important force shaping the structure of gut microbiota, much more important than genetics and other factors. So by working with a particular group of plants in China, it's called traditional Chinese medicinal food. It's published by Ministry of Health of China, and the plants in that list can be used as common food and also as medicine. And so we, over several years of research, we identified 2 groups of ingredients from these plants, polysaccharides, so non-digestible, fermentable polysaccharides, carbohydrates, and they can change the gut microbiota to a healthier structure because they are providing nutrients for some important beneficial bacteria, and also phytochemicals. And one class is alkaloids, like berberine, and has very dramatic effect on the total gut microbiota structure. It can essentially prevent obesity, high-fat diet-induced obesity in rats by daily gavage of a certain dosage of this berberine compound together with the high-fat diet. You prevent the animal from becoming obese and insulin resistant. And we showed that it has a profound effect on the gut microbiota structure. And the other compounds, like talked about by Ilya this morning, could be polyphenols and other compounds. They are protective. And so there are phytochemicals are a very rich source of factors which may be modulating or protecting our gut microbiota. So basically, by taking the human nutritional needs into consideration and also microbiome needs into consideration, and we developed a dietary scheme called Feed My Microbiome Diet. So I myself was the first volunteer. So over years, I lost about 45 pounds, okay, and over 1.5 years. And so this story was featured in Science 2012 in the microbiome issue. So if you are interested, you could look at that. And I would like to give you 2 examples from our research. The first one is identification of a particular human gut opportunistic pathogen which can induce obesity. So basically, we put this young man, 175 kilograms, BMI nearly 60, on this dietary intervention for 23 weeks. And he lost 51.4 kilograms. And he recovered from type 2 diabetes, from hyperlipidemia, hyperinsulinemia, hypertension. He was hyper everything before the intervention. And but after 23 weeks, and he recovered from all the metabolic problems except still some residual fatty liver problem. So basically, if you look at the inflammatory cytokines and all anti-inflammatory cytokines, you see elevation of inflammation. If you look at the endotoxin load as indicated by the surrogate marker lipopolysaccharide binding protein, it was significantly reduced after the intervention. And A1C reduced to normal range. And by using PCR DGG fingerprinting, we could generate fingerprints of gut microbiota at different time points. So at the baseline, you see 3 bands representing 3 major populations of bacteria, and they disappeared after a few weeks on the diet. And so after sequencing this the DNA eluted from these bands, and these bacteria identified as members of the genus Enterobacter. So there are 10 species in this genus. They are all sepsis-inducing, gram-negative. So the LPS on their cell surface is highly pro-inflammatory. And So this group of bacteria was about 1/3 of his total gut bacteria, but then disappeared quickly, and then he started to recover. So we suspect maybe this, the presence of such a high population of an opportunistic pathogen in his gut may be contributing to his metabolic problem. So then we isolated several hundred single colonies, and we co-migrate each single colony against the baseline microbiota fingerprint. So any single colony whose V3 region of the 16-RN gene can co-migrate to identical position to these 3 bands, we think this is the candidate we like. So eventually we isolated a bacterial strain called Enterobacter cloacae B29, and it was very high before intervention, quickly declined to non-detectable after a few weeks on a new diet, and then the host started to recover. So maybe this is the pathogen for his problem. So we inoculated this single pathogen into germ-free mice gut, and they can grow to a very high level, and you only need to inoculate once, and then they can just reproduce. So they can colonize germ-free mice gut to a very high level. But if you give high-fat diet, if you give normal diet, even though the bacteria is very high, nothing can happen. But if you give them high-fat diet, they develop, they have fully developed obesity, and fat pads significantly higher, and also insulin resistance, and fatty liver, and also the FF gene was inhibited to a low level by colonization of this pathogen in the gut, while in the liver, the genes which are required for synthesizing new fat, triglycerides from glucose, was significantly promoted by colonization of this single bacterium in the gut. So where Professor Gordon's group identified the gene level changes by the whole gut microbiota, we actually identified one single human gut pathogen which can do exactly the same. And interestingly, high-fat diet seems to be the environmental condition for this single pathogen can induce obesity. So no, if it's a normal diet, you have very low level of endotoxin in the bloodstream and no obesity. But if you give high-fat diet, you see a high level of endotoxin in the bloodstream and inflammation and everything, and eventually obesity and insulin resistance. So this this was reported in 2012, and seems to be the first single opportunistic pathogen identified by fulfilling Koch's postulates, which can cause obesity. And by working with, in the past 5 years, working with Professor Philippe Gerhard at INHA, the Agricultural Research Institution in France, and we move on to understand the molecular mechanism. And we actually identified a few more such pathogens, like this Escherichia coli strain. It was overgrowing in this baby girl's gut. She was 3 years old and almost 90 pounds. And so after put her on this dietary scheme for 3.5 months, she lost 16 kilograms and could lead a normal life. So we also showed that this single pathogenic strain can induce obesity in germ-free mice if you give high-fat diet. If you give normal CHO, it's no disease. So the second example I would like to give you is genetically obese children. It's called Prader-Willi Syndrome. So this is a very difficult form of obesity to deal with, because children with this genetic defect, after weaning, they develop a hypophagia. They feel hungry all the time, and you cannot satisfy them with any amount of food. And so it's a very difficult form of obesity to control. But accidentally, we found out that 1/3 of the mobile obese children in a nutritional intervention program, we worked together with Guangdong Women and Children's Hospital. And 1/3 of the mobile obese children in that program actually Prader-Willi syndrome patients. And they responded quite well to this dietary intervention, changing the gut microbiota. So like this boy reduced from 140 to 83.6 kilograms after 285 days in this program. And then after went home, he continued this intervention, and after 430 days, he came back for medical checkup, and he was 73 kilograms. So all our patients, mobile obesity patients on this dietary program, had no exercise, so just dietary intervention. And he recovered his insulin sensitivity, for example, to a very high level. And basically, so eventually we recruited 17 PWS children. They stayed in the hospital for 3 months, and 21 mobile obese children without known genetic reason, simple obese children, they stayed in the hospital for 1 month. So at the baseline and end of each month, you did a medical checkup for metabolic health and also collection of urine, blood, and fecal samples. And 1 month in the hospital, they lost about 10% of the initial body weight, and 3 months in the hospital, 20%. And they showed significant improvement in glucose homeostasis, lipid profile, and liver function, and also significantly alleviated inflammation in their system, and also reduced endotoxin load. And then we transplanted the before intervention gut microbiota and after intervention microbiota from the same individual to germ-free mice. And so before intervention gut microbiota, in the first 2 weeks, developed inflammation in liver and the gut. After the inflammation was gone, and they start to catch up with their body weight growth, but primarily by synthesizing new fat. So at the end of 2 weeks, the adipocytes are becoming very small, but after another 2 weeks, they become exceedingly large. So basically, the baseline gut microbiota had the capacity to induce inflammation and also promote excessive fat accumulation. But the after intervention microbiota from the same individual had no such effect. So this is just to show that diet-induced changes of the gut microbiota may have contributed causatively, at least partially, to the recovery of the metabolic health. And then we did a metagenomic sequencing of all the fecal DNA samples, inter-individual and intra-individual fecal samples. Each sample sequenced 80 million reads, and going through the bioinformatics pipeline, we identified a little bit over 2 million non-redundant microbial genes. And then we did the first round of co-abundance analysis at the gene level. So basically, we got a little bit over 20,000 co-abundant gene groups. And remember, each such co-abundant gene group may correspond to one DNA molecule, because all the genes in the same DNA, they should change their copy number exactly the same. So if they are highly correlated with their abundance across different samples, and then they can be grouped together and potentially are the genes from the same chromosome or same virus or same plasmid. But basically, because we are interested in bacteria, so we see 317 such CAGs containing more than 700 genes, and which are many are typical bacterial genes. So these 376 are potentially genomes of bacteria. And then we selected 161 from this because they are shared by more than 20% of the samples. So these are genomes of prevalent bacteria. And then we do CAG-specific assembly. So we were able to get 118 high-quality drafted genomes. When you get genome level, each high-quality drafted genome is either one single string or a group of highly similar strings. They share more than 95% of genes. And so when we get the genome level, the high-quality drafted genomes, we start to study their interaction from each other and also their relationship with the host. So we did the second round of co-abundance analysis at the genome level. And we were able to delineate this 118 genomes of prevalent gut bacteria into 18 functional groups. So members of each group, they increase or decrease together. So they can be considered as a guild in ecology. But if two guilds were connected with red line, they co-occur. They also co-occur. But if they were connected with the blue line, they co-exclude from each other. So now you organize 61 prevalent gut bacteria into such an ecological network. So you know who work with whom and also who compete with whom. And basically our diet can significantly promote this group of bacteria. 3 of them are in genus Bifidobacterium. And when they were promoted by the diet to a high abundance level, many other bacteria in these negatively associated groups declined. And many of these declined bacteria, they are potentially pathogenic or producing some metabolic detrimental compounds, such as indole or hydrogen sulfide. So we hypothesize that this group of bacteria may be the tall tree species in the gut forest. So when you grow them to a high level, and then they change the gut environment, and they suppress the pathogenic ones, and only allow some associated beneficial bacteria or neutral bacteria can grow. So starting from 2 million individual genes, we reduced to 376 bacterial genomes. And then we selected 161 prevalent gut bacterial genomes. And then we started their ecological interaction, delineate them into 18 functional groups. Because all the members in the same group, they increase or decrease together. So you can lump all the abundance of individual members to get the group level abundance. So now you get 18 variables. And then if you do correlation analysis with all the metabolic phenotypes, many start to show correlations. Like 3 showed a negative correlation, 9 groups showed at least 1 positive correlation with disease phenotype, but 6 showed no correlation. So now in terms of the relationship with the host, the blue arrowed ones seems to be beneficial bacteria. And the red arrowed ones may be potentially detrimental or pathogenic groups of bacteria. And the other uncorrelated bacteria, they may be neutral or commensal. And so by using this kind of data mining approach and high throughput sequencing, combining ecological thinking with the metadynamics approach and work at the gene level, and particularly at the genome level, you probably can develop a sound understanding, at least on the major members of the gut bacteria. And so this would form a very solid basis for future mechanistic studies. And I would like to maybe just very quickly go over this taxon-based analysis, because currently many researchers in the microbiome field sequence their microbiota and analyze at a species level, genus level, phylum level, whether there are any taxon significant difference between disease and a healthy sample. And I just would like to show that members of our guild can be from 1 phylum or from 4 different phylum. So their taxonomy seems does not play a role here in terms of who does what with whom. And in the same species, like Faecalibacterium prosnice, we assembled 9 genomes, and they are divided into 4 functional groups. So that means you need to understand the role of gut microbiota to each other and also to the host in terms of what they do, not what you call them, okay, not based on their taxonomy. And so gut bacteria work as guilds. So you need to identify who does what with whom, and then study them based on this ecological interaction. And I would like to also show you that in the same individual we followed, and we found in the first 105 days, there's a promotion of Bifidobacterium genus. But if you go down to a species level, only 1 species actually promoted significantly by the diet. And we isolated 5 strains from this species, And they shared most of their genes, and they had only a few dozens of genes unique to each other. So they are highly similar to each other. But if you look at the response, they still showed a differential response to the dietary treatment. So strain level understanding is the key. Otherwise, you are lumping different things together. You never can understand or how your diet, how your prebiotics, or whatever intervention you do, what kind of changes you induce, and whether that is good for the host or not, must go down to strain level, to genome level, to understand all these important questions. I would skip this story, and I'll finish with my classical joke. Okay. So basically, we are told by geneticists that our lifespan may be 120 to 150 years old. And so this could be our health status at any given time point. The most healthy lifespan would like this. So you remain healthy throughout most of your lifespan, and you die in the last week. Okay. And people with genetic defects can die prematurely very early. But most of us actually go a trajectory like this. So before 50 years old, we are okay. After that, we are going down the hill and eventually end up with in the hospital with one or several chronic conditions. But if the decline of our health after mid-age, 55 years old, and is due to the disruption of your gut microbiota, and then we have hope, right? So we can move back, because gut microbiota has a very high plasticity. It's manipulable. And our own genes is very hard to manipulate. And so we probably can move back to this region and maintain as long as we can, primarily by using nutrition, by using diet. So eat right, keep fit, live long, die quick. Okay? Okay.